Hi, my name is Kyle Savage. I do biology A-level. In this video we're going to be going over apoptosis. By the end of this video you should be able to say how apoptosis can act as a mechanism to change body parts, and that's practically it, because it's not that bad. In apoptosis, basically, there's quite a lot of history bits that you don't particularly need to know. I don't think you really do either, but they're nice to know. If you did history A-level, actually no, if you did history GCSE, I'm not sure about A-level, I don't know if it covered it. But if you did either of those, you might have a bit of a knowledge of the history of medicine. Yeah, I'm in my pyjamas, so sorry, I'm in my pyjama shirt, so had a long day. Might be able to hear the people next door, they're playing football, and by the sound of it, the older son is winning, so, and I don't think you'll be, my parents talking next door, but I don't think you'll be able to hear that on the video. Okay, so apoptosis, I am aware, all of the people that comment and inbox me, I'm pronouncing lots of words wrong, but I do say sorry for that. Even, I think I mentioned that in a video before, I'm saying them wrong, I'm sorry. But, yes, please stop, because I know, and I write it in the comments. Apoptosis is supposed to be pronounced apoptosis, but I think if you pronounce it how it's written, it kind of helps you to remember the exam, because at AS level, if you wrote something and you wrote it phonetically, they'll give you the mark. I'm not sure quite how true that is for A level. I think my teacher said it's not quite that lenient, but I still go with that theory. So if you can't remember how to spell a word, just keep saying it and write it literally how it sounds. Unless it's like glycogen, glucogen, you can't really get them wrong. You need to know the specific ones. So to start with, apoptosis. The simple dictionary definition is programmed cell death, and this occurs in all multicellular organisms. So it occurs in you and me, it occurs in your cat, it occurs in the woodlouse at the bottom of the garden, it occurs in all multicellular organisms. Cells should undergo about 50 mitotic divisions, and then they undergo a series of biochemical events, and that leads to an orderly and really tidy death, basically one that's planned, one that you know is coming. They don't just die randomly. This is a complete contrast to something called cell necrosis, which we don't need to know in depth about as much, but you should know that cell necrosis is cell death as well, but it's cell death that happens due to a trauma or something, and that releases hydrolytic enzymes, and yeah, it's basically a really damaging and bad cell death. We don't want that. I'm going to go over, there's quite a lot of a history about this, but I'm going to do that in the end, and just go straight through apoptosis, the scientific bits first. Well, basically, there's a sequence of events. There aren't that many, there's only 6, so you get off pretty lightly. Start off with, enzymes come in, and they break down the cell cytoskeleton. So, enzymes come in, break down the cytoskeleton, easy. Next thing that happens is that the cytoplasm condenses, because the cytoskeleton is broken down, the cytoplasm becomes dense, and all the organelles therefore become really tightly packed because it's so dense. Then the cell surface membrane changes as small bits called blebs form. Is it just me, or is blebs the best word that they've ever invented for use in science? You get some good ones, but blebs, that is a good word. So, the 3 things that happen so far is enzymes are come in and break down the cytoskeleton. The cytoplasm has become really dense, which meant the organelles get tightly packed. And finally, the cell surface membrane changes. Football's going right outside. And blebs form. Next, chromatin condenses and the nuclear envelope breaks, which means that the DNA breaks into fragments. The cell breaks into vesicles, and these are taken up by phagocytosis. Quick definition, do you know what phagocytosis is? You should do. If you don't, it's the endocytosis of large solid molecules into the cell. It's like when they go, it comes in. Cellular debris is disposed of and doesn't damage any cells or tissues. And it all happens really quickly. So that's basically it. The main points: enzyme breakdown cytoskeleton, cytoplasm becomes dense, cell surface membrane changes blebs form, chromatin condenses, nuclear envelope breaks, DNA breaks into fragments, and the whole cell is broken into vesicles, which is taken up by phagocytosis. So it's not that hard, it's good. Apoptosis is a really good programmed cell death that has to occur in the body. They used to think that cells were immortal, but they're not, because that wouldn't work. but we'll look at that in a second. So how is it controlled? Obviously this can't happen just on its own. It does have to be under some control. Well, the process is controlled by a diverse range of cell signals. This is literally read from the book at the moment because I haven't really got a better way to explain it. And some of these come from inside the cell and come from outside the cell signalling. These signals include cytokines, which are made by cells in the immune system, also hormones, growth factors, and nitric oxide. Nitric oxide can induce apoptosis. This induces it by making the inner mitochondrial membrane, which remember is folded, makes that more permeable to hydrogen ions, and that dissipates the protein gradient. Change that. Proteins are then released into the cytosol, and these proteins bind to apoptosis inhibitor proteins, and they allow the process to take place. So if we go over how it's controlled again, the process is controlled by cell signals from outside or inside the cell. These include cytokines made by cells in the immune system, can also by hormones, growth factors, or nitric oxide, which can induce apoptosis by making the inner mitochondrial membrane more permeable to hydrogen ions and dissipates the proton gradient. And then the final bit is proteins are released into the cytosol, and these bind to apoptosis inhibitor proteins and allow the process to take place. Sorry, I know it's really bright, but I'm finding it hard to see the thing. That is good so far. So now you know how apoptosis is, how it occurs, and how it's controlled. But the definition we got at the beginning, what you were going to know by the end of the video, was how it can act as a mechanism to change body plans. And apoptosis. So how is apoptosis linked to development? Now, it's an integral part of plant and animal tissue development, and there's so much division, extensive division and proliferation of a particular cell type, followed by pruning through programmed cell death. The excess cells shrink, fragment, and are phagocytosed so that the components are reused and no harmful hydrolytic enzymes are released into the surrounding tissue. Quick catch up. Do you remember when hydrolytic enzymes are released? I'll give you a clue. It is bad. It's actually a bad cell death. You're right. It's cell necrosis. So apoptosis is really tightly regulated. Like, can't stress how tightly regulated. It's really and different tissues use different signals for inducing it. It weeds out the ineffective or harmful T lymphocytes during the development of the immune system. Now we're going to look at an example. During limb development, apoptosis causes the digits, which are your fingers and your toes, to separate from each other. So when you're in, when a little baby, little embryo fetus thing is being born, it's born and it's got it's still got a skin between the fingers basically. These aren't separate at all. If you look in, it's like this. So you've got like little flipper things. Apoptosis means little flipper things turn into little hand things, which you can use for stuff. They're generally very useful. In children between the ages of 8 and 14 years, 20 to 30 billion cells per day undergo apoptosis. That is a lot. Of course, there's a lot more in your body, but think 20 to 30 billion cells per day undergo apoptosis. In 1 year, this equates to a mass of cells equivalent to the total body mass. So the number of cells that have undergone apoptosis is like the number of cells that is the mass of an 8 to 14-year-old. In adults, even more, 50-70 million cells per day apoptose. Apoptosis is really key in development in your body. The rate of cells dying should balance the rate of cells produced by mitosis, obviously, because you still want your body to be there. You don't want it to just die, because if 50-70 million cells died per day and went put back into your body, then you'd just disappear. If the rates are not balanced enough, not enough apoptosis leads to the formation of tumours. And also, too much apoptosis leads, as I said, to cell loss and degeneration. Degeneration, I said that? No. Degeneration. Yeah, degeneration. If you want an example of what can go wrong in apoptosis, something that's quite common is webbed fingers or webbed toes. I think you generally get webbed toes more. I haven't actually seen that many webbed fingers, I guess. I think webbed fingers are kind of a lot tricky to live with in webbed toes, so that can be surgically done. But webbed toes are really common. I've known quite a few people with them, and basically that's it's called syndactyl. I think that's how you say it. Apologies if it's not. And that's because of lack of apoptosis. Apoptosis hasn't fully been completed, so your toes are joined. If you want to look that up, just literally something like webbed feet or syndactyl, which is spelled Syndactyly. Yes. How do you spell it? Syndactyly. Syndactyl? Yeah. I read it anyway. And it's really interesting actually. Yeah, so look that up. So now we've gone over the apoptosis bit, we have to look at the history of it.